Start, sir.
Good morning, dear students. Welcome back to another live webinar class of zoology subject. Dear students, let us begin this class by continuing the last class. In the last class, we have started the chapter excretory products and their elimination for second year students, isn't it? So we will continue that class. This is the part 2 of that chapter. Okay. Let's see what we have learned in the last class. Let us recapitulate what we have learned from excretory products and their elimination, which is unit 2. We have started learning about the system, that is the excretory system. There you studied the excretory organs, which are kidneys, isn't it? The main organ is a kidney, and there are some associated organs also, like ureter, urinary bladder, renal vessels, isn't it? And when you observe one single kidney in detail, we have seen the structure of it, and there are two parts internally: the cortex, which is the outer part, and the medulla, which is the inner part, isn't it? And in the medullary part, there are medullary one single medullary pyramid. If you enlarge it, you find this kind of structure of this medullary pyramid contain the micro function. In the last class, we have studied LSS and the structure of nephron. In the nephron, what structures you find? You find the Malpighian corpuscle, which is the main important organ, first organ of the Malpighian corpuscle contain this glomerular filtration body, which is glomerulus, and U-shaped structure called Bowman's capsule, and PCT, which is the proximal convoluted tubule, and Henle's loop, the long tubular structure you are able to find here. This is the Henle's loop, and there is a DCT, which is distal convoluted tubule, and collecting duct finally. These are the important parts of the nephron. Today, in the process of learning about the formation of urine, we will be studying the biochemical process which take place in the structure called nephron. Okay? So for this learning, you must be aware of the structure of nephron. That's why I have shown here. Let me explain you again. The first part of the nephron, you can see on the slide, is Malpighian corpuscle. It contains two parts called glomerulus and Bowman's capsule. The immediate next part is PCT, which is proximal convoluted tubule, and the third part is DCT, which is distal convoluted tubule, right? And the long tubular structure which is extending to the medullary region is called as Henle's loop. It's also called loop of Henle. Okay, and this tubular network which you are able to see around this loop of Henle is called vasa recta. Okay? The final part is called the collecting duct. This is the structure of nephron. Okay, let's see now about the formation of urine. Okay, let's start. Concepts that we are going to cover from this class is glomerular filtration, selective reabsorption, and tubular secretion at proximal convoluted tubule, and same reabsorption and secretion at Henle's loop, and reabsorption and secretion at DCT, which is distal convoluted tubule. And we also learn about the what happens in the collecting duct also, and we learn about mechanism of concentration of urine, because the concentration of urine keeps on changing while it is passing from the renal tubule. And finally, we learn counter current mechanism, which is most important concept. Okay, so this is a little typical class. So to understand, little typical subjects are there, concepts are there. That's why I request you to listen very carefully. Okay, so this is the again introduction. Excretion. We learn about excretory system and biochemical process, which is involved in the excretory process. In the excretory system, we learn kidney, ureterus, urinary bladder, urethra, and the biochemical process. We learn about 3 steps. The first step is glomerular filtration, second tubular reabsorption, third tubular secretion. This is the complete picture of what we are going to learn from this chapter. We have already studied this, that's why I'm going a little faster. So let's learn the first step, which is the glomerular filtration. Okay, this is the first step in the formation of urine. What is mean by the urine formation? Actually, how does the urine is formed? By the filtration of the blood. Okay, I repeat, by the filtration of blood, urine is formed. This filtration of blood take place in the kidneys and the ultrastructure. You can find the nephron, and the nephrons are the sites of the formation of urine. Okay, let's see. The first step is glomerular filtration. The name itself says that there is something which is getting filtered. Okay, so it is the filtration of blood from glomerulus to Bowman's capsule. Now you must know what is mean by glomerulus and Bowman's capsule. Beside you can see the diagram. In the diagram you can see the capillary, blood capillary structure. This blood capillary network of blood capillaries is called the glomerulus, and the U-shaped structure which you are able to see, which is in white colored, it is called the Bowman's capsule. Okay, so it is a passive process. It does not require any energy for the filtration. Okay, why does it, how it can be filtered without any energy? You must be getting doubt. So for this we need we have some pressures which is applied. Okay, with the help of these kinds of pressures, the filtration is able to happen. What are the pressures helps in the formation of urine? Let's see. The first kind of pressure is hydrostatic pressure in the glomerulus. Hydrostatic pressure is applied by the liquids. Okay, so liquids produce this hydrostatic pressure. This pressure, when you measure it, it is around 60 mmHg. Okay, 60 mmHg. The next pressure which is applied in the glomerulus is the glomerular colloidal osmotic pressure. Okay. This colloidal osmotic pressure is created by the concentration gradient. The differences in the concentrations can produce some kind of pressure. That's called osmotic pressure. Okay, this is around 32 mmHg. The next pressure which is applied is the Bowman's capsule hydrostatic pressure, which is 18 mmHg. Okay, so these are the 3 kinds of pressures which are applied, and finally the net pressure is 60 minus 32 plus 18. Now we must understand which pressure works at which direction. Okay, if we know this, we can subtract from higher pressure to the lower pressure. Let's see what are the changes happen here. Let me show you how does it happen. Okay, and wait 1 second. Okay, you can see here the first pressure, which is hydrostatic pressure in glomerulus, which works against to the Bowman's capsule. Okay, so this will work against to Bowman's capsule, and glomerular colloidal osmotic pressure works against to the glomerulus, and the next one is Bowman's capsule hydrostatic pressure works against to the glomerulus. So here the 60 minus 32 plus 18 is equal to finally we get 10. Okay, so this is the finally around we get 10 mmHg. So this is the final pressure which is applied on the filtration. So this 10 mmHg is the reason for the filtration process. Okay, let's learn further. Okay, you can see the cascade of the urine transport. So blood in the glomerulus, there are small hole-like structures in the glomerulus. They are called fenestrations, and there are podocytes which are existing in the glomerulus. And through the podocytes, the urine comes into the Bowman's capsule. Hence, the process is called as ultrafiltration. Okay, let's see further. The ultrastructure of Malpighian body. You can see here, this is the kidney, and if you take the glomerular Malpighian corpuscle, it looks like this. And here you can see glomerular structure. Okay, so if you observe the network of the capillary structure, you find this kind of structure where you can find the podocytes. You can see here, these are the podocytes. Okay, these podocytes have food processes, and there are fenestrations in endothelial cells which show the passage of the urine. Okay, so the blood gets filtered and urine comes down. Okay, through with the help of these podocyte and fenestrations. Okay, so through these it will come out. Okay, let's learn further. So whatever the urine formed out of this Malpighian corpuscle is called the primary urine. You can see here, this is the glomerulus, the U-shaped structure called Bowman's capsule. The blood gets filtered, and the primary urine is formed. This primary urine. Okay. Primary urine composition, we must know what is present in the primary urine. Primary urine contain carbohydrates, albumins, globulins, water, nutrients, and minerals and salts. So there are useful substances also in the primary urine, isn't it? You can see here, carbohydrates are useful for us. They produce some energy. Albumins and globulins are also important for us. Water is also, to some extent, it is required for the body. Nutrients are also required, and minerals are also required. So there are useful products and also the harmful products in the primary urine. Creatinine and urea. Okay, creatinine and urea, uric acid are also gets filtered by this glomerulus. Okay, so in the primary urine contain useful substances, also the harmful substances. So what we need, the useful substances must be returned, they must be reabsorbed. Okay, that is the next process, which is tubular reabsorption. You can see here, except the proteins, all constituents are there. So it's hypotonic to the cortical fluids. Okay, it is less concentrated than it. And you can see here, around 1100 to 1200 ml are filtered in 1 minute. That means more than 1 liter is filtered per 1 minute. So this is the fastness of the glomerulus. That means all the nephrons which are there, all the nephrons, there are millions of nephrons all together. So they can make 1.2 liters of urine, 1.2 liters of primary urine, okay, per minute. Glomerular filtrate, you can see 125 ml per minute, and it is filtering 180 liters per day. So you can say like this, that our kidneys capacity is 180 liters of urine can be produced per day. But what we do, we only excrete 1.5 liters per day, isn't it? So what is happening here? You can see 99% of the primary urine is getting reabsorbed. Okay, so our kidneys can filter 180 liters. One can make 180 liters of urine per day, but all the urine, if we excrete, what happens? We lose all the water in the body, and in the process there will be osmotic gradient created and we eventually die. So to protect us, to maintain the water and ionic balance, we reabsorbed all the substances, all the useful substances along with water. Okay, so usually we excrete 1.5 liters per day, and 99% of the primary urine is getting reabsorbed. Let's see the process of reabsorption and secretion. You can see here about 85% of primary urine is reabsorbed by PCT, which is and descending loop of Henle. So proximal convoluted tubule, PCT full form, and descending loop of Henle are important for the reabsorption purpose. Okay, mostly it is PPT, sorry, PCT, which is obligatory and mandatory. Rest of the reabsorption is regulated based on necessities. If we have less water in the body, if the water content is so less, then there will be hormonal mechanism which initiated, and these hormones, what do? They involve in the process of reabsorption. Okay, they they stimulate the DCT, which is distal convoluted tubule, for the reabsorption process. Let's see what are the substances reabsorbed. Based on the amount of reabsorption, the contents are classified into three types. The first type is high threshold substances, which are mostly reabsorbed. Essential and efficiently reabsorbed substances comes under the high threshold substances. They are glucose, amino acids, vitamins, and some salts. Okay, next, low threshold substances. They are absorbed very little amounts. Example, urea and uric acid. The next is A threshold substances. That actual excretory products are called as A threshold. That means they do not get reabsorbed. Okay, they don't get reabsorbed. Example, creatinine. Okay, creatinine is the completely excretory product. Okay. Let's see further the reabsorption and secretion. And this is the overall picture of reabsorption and secretion. You can see here the red colored substances that are given here are called reabsorbed substances. Okay, in the PCT, this is the part of PCT. You can see here, this is PCT. This is the part called Henle's loop, and this is DCT, which is distal convoluted tubule, and finally this is the what is this? This is the collecting tube. So these are the different parts, and at different locations there are reabsorption taking place. Now we will learn at each level, at each part, how the reabsorption takes place. Okay, dear students, let's see. First of all, the PCT, that means proximal convoluted tubule. This PCT is lined by cuboidal epithelium with the brush border. This brush border in the epithelium indicates the more reabsorption. Okay? So in the small intestine, you can see the villi structure, isn't it? For what? For the increasing of the surface area, for efficient absorption. The same way, PCT contain brush bordered cuboidal epithelial cells, and these helps in the process of absorption. Okay, next, 70 to 80% of the electrolytes and water are reabsorbed here. Next, Na, which is sodium, is actively transported into cortical interstitial fluids. This transfer of positive charge drives the passive transport of Cl minus, glucose, and amino acids. Okay, NaCl is getting, or you can see here, creatinine, antibiotics, diuretics, and uric acid. These are getting absorbed, and whereas reabsorbed substances are NaCl, K plus, and water, bicarbonate, glucose, and amino acids. These are getting reabsorbed. PCT maintains pH and ionic balance by selective secretion of hydrogen ions and ammonia into filtrate and reabsorption of copper and HCO3 from it is spelled wrong, it's copper. Okay, let's see here. First, you must know what is the where the filtered substances go and where the reabsorption takes place. Let me tell you, reabsorption means coming out of the renal tubule. It is called reabsorption. Okay, if something is getting into it, okay, it is getting in. If something is getting into it, that's not called reabsorption. That is called secretion. Okay, you can see here, suppose if something is getting inside of the PCT, that is called secretion. That means substances are excreted, so they are secreted into the PCT. Reabsorption means coming out of the renal tubule. Coming out is called reabsorption. Getting in is called as secretion. Okay, you must be clear on these parts. That's why I'm repeating again and again. So here, creatinine, antibiotics, diuretics, and uric acid are getting secreted. They are going to get eliminated. And NaCl, K plus ions, water, bicarbonates, glucose and amino acids, these are getting reabsorbed. That means they are going to come into the body. Okay, hope you are able to understand this reabsorption and secretion. The term reabsorption and secretion, you must know these two terms. Reabsorption means we are taking in for the body. Secretion means we are excreting. Okay, let's see further. Movement of water occurs by the process of osmosis. Do you know what is meant by osmosis? Osmosis is the transfer of molecules from its higher concentration to the lower concentration, from high water concentration to low water concentration with the help of a membrane. Okay, a semi-permeable membrane. It's called osmosis. It is different from diffusion. Diffusion means transport of substances from high concentration to the low concentration. There is no semi-permeable membrane there, but in the osmosis there is semi-permeable membrane, and the movement of substances from high water concentration to low water concentration. Let's learn further about what happens in the Henle's loop. First of all, you must know where the Henle's loop present. This U-shaped structure you are able to see on the slide is called as Henle's loop, which is also called loop of Henle. So Henle's loop contain two parts in it. The first part is descending limb. That means it is going down, isn't it? And the other part is ascending limb, which is coming upwards. You can see here on the diagram. And reabsorption is very minimum in the Henle's loop. You can see descending limb is permeable to water and impermeable to electrolytes. Hence, reabsorption continues and filtrate concentration gets increased gradually as it moves downwards. You can see here the H2O molecules, water is getting out, isn't it? Ascending limb proximal segment is thin, in which NaCl diffuse outside, and interstitial fluid also gets passively excreted. The distal segment is thick, and which is where the NaCl actively pumped out. The ascending limb 20 ascending limb, and it is impermeable to the water molecules. Here you can see as it comes down in the descending limb, water molecules are moving out. When water molecules are moving outside of the limb, what is happening? The concentration of substance is getting increased, okay, as it moves down. Let's see what happens in the PCT and the DCT. Sorry, not PCT, it's DCT. In the distal convoluted tubule, cells are shorter and lack microvilli, which indicates less reabsorption. In the PCT, we have seen the microvilli, structure of microvilli, but in the DCT there are no microvilli. Okay. Next you see the conditional or facultative reabsorption takes place here. So DCT only reabsorbs whenever it is necessary for the body. So this signal is got by the hormone called aldosterone and also the ADH, antidiuretic hormone. Okay. Reabsorption of water is dependent upon several conditions. It is regulated by ADH, antidiuretic hormone, for maintenance of pH and sodium potassium balance. So a small correction here. What you must do is this PCT you should convert it as DCT. Okay, it is actually about DCT. Okay, dear students, let's learn further. Reabsorption and secretion in the collecting duct. It's a long tube that carries filtrate through the middle area to the renal pelvis. Okay, it's a long tubular part. Water reabsorbed slightly in this segment and it allows the passage of small amounts of urea to the medullary interstitium to keep up its osmolarity. Okay. If something is getting into the medullary interstitium, it means it is entering to the blood vessels. Okay, the urine in the CD, that means collecting duct, is hypertonic to the plasma of blood. Okay. You can see here the urinary secretion. The urinary secretion is equal to glomerular filtration minus tubular reabsorption plus tubular secretion. Okay, this is an equation, final equation for better understanding. Let's see what happens in the mechanism of the filtrate. How concentration of filtrate gets changed. Loop of Henle and vasa recta plays a very important role, very significant role in producing the concentration of the urine. What is meant by concentration? First, you must know this. If the amount of more in a solution, if the solute concentration is more in a solution, it's called a concentrated solution. So increase of the solute concentration, increase the concentration of that solution. The flow of renal filtrate into limbs of Henle's loop is in opposite direction. Here in the diagram you can see, isn't it? So the flow you can see, it is in the opposite direction. In the descending limb, the renal fluid is going downwards, and the ascending limb, the renal filtrate is coming upward. So the flow of this urine is opposite to each other, isn't it? So this is nothing but the counter. Okay, counter means nothing. The name itself says counter means opposite. Okay, counter. Current means the flow. So here the counter current pattern you are able to see. Same here in the vasa recta also, you can see afferent arteriole, efferent arteriole, the blood flow is opposite to each other, isn't it? Right? This is also opposite to each other. So this opposite flow of the blood, opposite flow of the filtrate, okay, this will create counter current pattern. Let me tell you, counter means opposite, current means the flow. So here the blood and also the urinary filtrate both are flowing in an opposite direction. This opposite flow is called as counter current pattern. Okay, dear students, hope you have understood. So in this you can see the concentration variation also. Here in the fourth point you can see it helps in maintaining the increasing osmolarity towards in medullary interstitium, that is 300 osmolarity in the cortex to about 1200 osmolarity in the medulla. Okay, which is mainly found by the transport of NaCl and urea. NaCl enters descending limb, making it more concentrated. Right? and concentrate, more concentrate. NaCl gets out of the ascending limb, making it so dilute. So what happens in the concentration gradient, we will be learning in the next slide. Don't worry. You may be getting confused, a little confused about it. We will clarify everything. Okay. Let's see further. Let us delete all drawings which are on the board. Okay. Osmolarity changes. What are the osmolarity changes taking place? In the beside, you can see the structure of nephron. And now I will be explaining you how does it happen. Dear students, look at here. Here the concentration of the filtrate is 300 osmolarity. Okay. This 300 osmolarity, as the fluid is moving downwards, its concentration is getting increased. You can see here the concentration is 300 osmolarity, and here you can see the concentration is 400 osmolarity, and as it moves downwards, the concentration is getting increased to 600, 800, 1000, and finally it is reaching to the 1200 osmolarity. Okay, so in the cortical region, the concentration osmolarity is less. As it moves down, the concentration is getting increased. How does it getting increased? That's the question. So see here, because of the outward movement of H2O, here I have given, because of the outward movement of H2O, that means water, the concentration of the solution is getting increased. Let me tell you what is meant by the concentration. Here osmolarity, the definition is given, you can look at that. The concentration of a solution means it is calculated by the total number of solutes per liter. What is meant by solute here? I will tell you the example. Suppose we have taken a glass of water and we have added a salt to it, a spoon of salt to it. So here the water is the solvent and the solute means the salts that we are going to add. The spoon of salt we have added, that's called solute. Okay. So final product is called as solution. When salt is mixed with the water, that's called a solution. Okay. So now you might have understood the what is meant by solvent, solute and solution. If the solutes are high, if we add 10 spoons of sugar or 10 spoons of salts in one glass of water, its concentration is very high, its osmolarity is high. Why it is high? Because the number of solutes per liter is high. Okay. Amount of solutes decides the concentration. Amounts of solutes decides the osmolarity of a solution. Okay. So here, when you are removing the water content from a solution, what is happening? Finally, the concentration of that substance will be increased, isn't it? That's what happening here. In the PCT, the concentration of filtrate is 300 osmolarity. Due to removal of H2O, due to removal of water in the descending loop of Henle, what is happening? The concentration is increased to the 1200 osmolarity. Why it has to increase? Because the medullary osmolarity is high. That's why it has to change according to the medulla. That's why for making the balance, it has increased its concentration. And this water which is gone out, it goes into the blood vessel, that is vasa recta. Here you can see the two structures you are able to find. This is called vasa recta and this is the renal tubule. Okay, remember two things are there, vasa recta and renal tubule. Here we are taking renal tubule, the part of the renal tubule called Henle's loop. And the second one you are able to see at the left side is vasa recta. Vasa recta means blood vessel, okay, arteriole. So whatever the H2O coming out of the PCT, it is getting into the getting into the arteriole. It is getting into the blood vessel. Okay, so renal tubule excrete, renal tubule getting reabsorbed, water is reabsorbing from the renal tubule and entering to the blood vessel, which is vasa recta. Okay, so remember only one thing, that as the filtrate moves downwards, the water, due to elimination of the water from the tubule, the concentration of the filtrate is getting increased from 300 osmolarity to the 600 osmolarity. You can see here you can see the next point here, the transport of substances facilitated by special arrangement of loop of Henle and vasa recta called counter current mechanism. Just now I told you, in the descending loom, the substance is coming down, and in the ascending loom is going up, and the same opposite thing is happening in the vasa recta. Blood is coming down and going up. So the flow of the filtrate and blood is opposite to each other. Since it is opposite, it's called counter current fashion, and this counter current fashion and the concentration changes in the counter current fashion are called as counter current mechanism. Okay, we will see the next structure, so you will get a proper idea regarding this. Before learning this, let's see another structure finally. Wait 1 minute. Okay. Observe this structure. Let me open another diagram. Yeah, this diagram you can see here. So reabsorption, you can see here, what are the substances getting reabsorbed. So the NaCl, the PCT, NaCl, K plus ions, water and glucose, amino acids, these are getting reabsorbed from the primary urine, and the DCT, you can see the water is getting reabsorbed. So this is the main substance, water is getting reabsorbed. Here the water, NaCl and glucose molecules are getting reabsorbed. Red colored structures are reabsorbed. Okay, Mg and Ca, calcium and magnesium are getting reabsorbed. Here also calcium and magnesium getting reabsorbed. NaCl is moving out in the ascending limb of loop of Henle, hence the concentration gets increased. In the next class, we learn about how the concentration changes take place. In the distal convoluted tubule, you can see NaCl is moving out, calcium ions are moving out, and water is also getting reabsorbed. And what are getting secreted? K plus ions and H plus ions, and urea is getting secreted. So this is the collecting tubule which takes it to the renal pelvis, and finally it forms the urine. Okay, so I will explain you again. I will give you a brief explanation about counter current mechanism also once again. Let me show you this picture. Dear students, you can see on the picture. So this is the structure of nephron, and this is the PCT, which is proximal convoluted tubule, which has 300 osmolarity, and its concentration getting increased as it is moving downwards. Why it is increasing? Because of the removal of water. Okay, if the water is removed from the solution, the concentration of that solution will be increased. Okay, so it increased to the 1200 osmolarity. As it is moving upward, there are other changes happening here. What is happening? NaCl is going outwards. You can see here. Since here the water is not moving out, but the salts, NaCl, is moving outward. Since the removal of the solutes, the concentration of a solution will get decreased. The same way happening here. Due to removal of the solutes, the concentration of the filtrate is getting decreased as it is moving upward. Here what is happening? The solutes are not getting removed in the descending limb. Solutes are not getting removed. Water is getting removed. That's why concentration increased. As it is moving upward, the solutes are removed. Hence, the concentration came to again 200 osmolarity. So this kind of concentration changes happen in the uh, structure of nephron. So these concentration changes and counter current flow. This will create counter current mechanism. You can see here the other structure called vasa recta. So here the blood is moving from down, from left to right to left. Here the filtrate is moving from right to left. Okay, so this is opposite in direction. So the concentration in the blood vessel also gets increased, like 300 to 1200 osmolarity as it is moving to the medullary region. Okay, why it is increased? Because whatever the NaCl substances removed by the ascending loop of Henle is entering to the vasa recta. For this reason, since the solutes are added to the blood, the concentration of the blood is getting increased. Okay, as it is moving upwards, what is happening? It is added with the H2O. Water is added to it. Okay, whatever the water removed by the descending loop of Henle, these water molecules are getting added. Let me explain you again. The NaCl which is removed by the ascending loop of Henle is added to the blood vessel. That's why concentration is getting increased to the 1200 osmolarity. The H2O molecules which are removed by the descending loop of Henle is added to the vasa recta. Okay, that's why the concentration is again getting decreased to the 300 osmolarity. So this is the counter current mechanism which exists in the process of filtration or the process of formation of urine. Okay, dear students, osmolarity changes. These are the osmolarity changes. Let's now see what is the assignment for you for today. If you have any doubts regarding this counter current mechanism, dear students, consult your subject jail and get the clarification done. Okay, so watch this video again and again so that you can understand. Okay, next assignment is write about glomerular filtration. Explain the process of urine formation. Process of formation of urine. This is a long answer type question. Learn this 8 marks question. And the third question is write a note on counter current mechanism. The name itself says counter means opposite, current means flow. Okay, so here the blood flow and the urine filtrate flow, both the flows are opposite to each other. Descending limb and ascending limb. The flow in the descending limb and flow in the ascending limb also opposite to each other. This opposite flow of the substances is called counter current mechanism. Okay. So this is about today's class. In the tomorrow's class, we will continue this chapter. Thank you, dear students. Thank you, madam. Madam, the class is ended.
Okay. Thank you, Rajkumar sir. You